Welcome everyone, and thank you for virtually joining us for the latest version of the Variant Effect Seminar Series. My name is Adrini, and I'm a member of the VASC Organizing Committee, together with Ian, Diego, Mariano, and Ohana. These seminars are made possible with support and a lot of help from Lara Muffley and Alex Hopkins, among several others, and our sponsors are the Atlas of Variant Effect Alliance. As a reminder, we are always on the lookout for potential speakers for future seminars. And if there's anyone you'd like to see presenting, or if yourself would like to present your work, then feel free to submit a speaker nomination form. You can find the QR code here and the links in the website. And as usual, before starting the presentations, I have a few housekeeping items to mention. So each talk is around 20 to 25 minutes, but there should be some time for a few questions at the end. So please post your Q&As, your questions at any time in the Zoom Q&A here, and we will read them out to the speaker at the end. And if you have additional comments or additional questions after the end of the seminar, and you are an AVE member, you can go visit our seminar series Slack channel and post your comment there, and we can have a discussion. And finally, we are posting on our Twitter and or X many updates, so be sure to follow us there. You can catch the seminar today, and previous presentations that are recorded in our affiliated CMAP SEGS YouTube channel. You can find the link here and also in our website. Don't worry about these URLs. I also post them in the comment chat. And all the details of the seminar series can be found in our website, including a regularly updated schedule of upcoming talks. And lastly, for the last housekeeping item, this is rarely an issue, but as a reminder, please be considerate in posting comments or questions. We expect everyone to be respectful and use welcoming and inclusive language. And finally, we can get started with our presentations of today. And first, you're going to have Ian Hoskins and then Gabriella Stevens. And Ian Hoskins has a Bachelor's of Science in Integrative Biology from the University of Colorado at Denver. And he worked with biotechnology at Enzymatics Inc. and Archer DX Inc., developing genomic assays and software for clinical cancer research. And now Ian is in the University of Texas at Austin in Dr. Ken Cenik's lab, and he is going to talk about integrated multiplex assay of variant effects and revealing gene expression mechanisms of catechol O-methyltransferase. And later we're going to have Gabriella Stevens talking about phenotypic landscape of the receptor tyrase kinase. So you can go ahead, Ian, and start your presentation. Perfect. You need to put the presenter mode now.
Sorry, I had to get my microphone on. Can everyone hear and see me?
Yes, go ahead.
Okay. Thank you. My name is Ian Hoskins. I'm a member of John Jenik's lab at UT Austin, where we study translational control. And today I'll be talking about integrating several multiplex assays to profile variant effects in the gene COMT. So as we all know, annotation of rare genetic variation is a serious challenge. This is why we have the Atlas of Variant Effect Alliance. And really the key here is how do we link rare genetic variation to phenotype. So it's estimated that about 50 other people on the globe share the same de novo genetic variants. And by de novo, I mean these are variants that arise during gametogenesis or post-zygotically, so not inherited through the germline. And as an example, many years ago, I actually had exome sequencing done for myself. And here I'm just showing for my variants that are less than 1% in frequency, how they are basically stratified by the predicted consequence. And so you can see that a relatively large proportion of the variants I have at low frequency are these variants of uncertain significance. Now, previous multiplex assays have really focused on variant effects on protein abundance and protein function, as well as organismal fitness. And a few other studies have really looked at the effects of variants on RNA abundance and decay. But to date, no multiplex assay has been developed to look at translation specifically. And we know that translation is important for protein abundance. It's also important for RNA abundance by sometimes leading to RNA decay. And so it's critical to understand how variants might impact translation in addition to RNA and protein abundance. And so really our central question in the lab is how do coding sequence variants alter translation and more broadly post-transcriptional gene expression? Now, we've chosen COMT as a target for multiplex assay because there's some prior literature suggesting variants in this gene may impact translation, and I'll get to that in a moment. But really, kind of the biology of COMT is it's a methyltransferase that will attach a methyl group to a number of important neurotransmitters. So all of these neurotransmitters have this backbone catechol structure, and COMT will add on a methyl group to this backbone to catabolize these compounds. It also deactivates catechol estrogens, which are known to be genotoxic. And by virtue of these functions, variants in COMT have been known to associate with neurological disorders as well as contribute to risk of cancer, specifically in breast and ovarian cancers. So it was previously found that certain common population SNPs in COMT that form a number of haplotypes may impact RNA structure. And so Knackley et al. In 2006 showed that 3 of these haplotypes, termed the low, average, and high pain sensitivity haplotypes, differ in their predicted alteration of RNA structure. And they further showed that these haplotypes led to decreased protein abundance and enzyme activity with higher amounts of structure. So you can see that this high pain sensitivity haplotype led to a lower protein abundance and enzyme activity. And so this was interesting. We have, and I should also note that many of these variants are silent variants. So yes, they might impact protein abundance. I think there's one that is a missense mutation, but the others are all silent variants. So we were really interested in this, and we wondered, do other rare COMT variants similarly alter RNA structure with consequent impacts on gene expression? So here I'm showing the predicted RNA structure in the early coding region. So exon 2 is the 5' UTR, and exon 3 is the first coding exon. And so there's relatively high probability structure in this early coding region. Oops. I should also mention that COMT is produced in 2 different protein isoforms. So there's a membrane isoform and a soluble isoform. And so we sought to profile rare variant effects in this early coding region, as highlighted with this pink line. So to do this, we first cloned COMT with its exon 2 5' UTR coding region and a C-terminal GFP tag, as well as a bicistronic iris M cherry. And this allows us to measure both protein abundance and a proxy for RNA abundance. And then we had variant libraries synthesized where we generated all silent and missense and 1 nonsense codon at each of 58 codons in the early coding region of COMT. And then we stably expressed this variant library using the improved landing pad cell line. And we generated 4 replicate cell lines per ROI. And ROI is a region of interest. So you can see we have 8 total cell lines, 2 ROIs, and 4 biological replicates. And then we performed a number of assays to read out the effect of these variants. So for instance, we sequenced the DNA, we sequenced the total RNA, we sequenced DNA after sorting cells by flow cytometry, and we further conducted polysome profiling, which allows us to read out translation. And so I'll walk through each of these assays in order. I'll also mention the analysis we've used is a custom software we built in the lab called SatMut Utils. This stands for Saturation Mutagenesis Utilities. And this is available online on GitHub if you'd like to use this for readout of your multiplex assays. So first, we looked at how COMT variants are impacting RNA abundance. So again, we sequenced the genomic DNA and the total RNA, and then did a targeted library preparation using an amplicon method. And finally, read out variant frequencies using SatMut Utils. So as a quality control, we would expect that nonsense variants would be decreased in RNA abundance by triggering nonsense-mediated decay. And indeed, we found nearly 90% of the nonsense variants we targeted were down at the level of RNA. We also found a number of missense variants and a single silent variant that were increased in RNA abundance. And I'll just mention this effect size that I'm showing is derived from ALDEX2 analysis. And these effect sizes are similar to log fold changes, but they're more robust as they rely on a non-parametric resampling strategy. So if you'd like to learn more about that, you can check out ALDEX2. So plotting this in a variant effect map for the 2 regions of interest, we found that, again, the nonsense variants were decreased in RNA abundance uniformly. Notably, we discovered this hotspot in region 1 at the H62 and V63 codons, where nearly all silent and missense changes led to increased RNA abundance. We also noted differences between the regions. So you can see in region 2, many variants actually led to increased RNA abundance. So we're really interested in this hotspot because there's such a strong signal, and we're curious what potential mechanisms might explain that. So we looked into a number of mechanisms. First, I'll mention that this H62 codon was previously identified as like the common population SNP at this position led to increased protein abundance in the context of this haplotype. That was concordant with this prior literature. And we looked at a number of mechanisms that might explain this. So codon stability coefficients, tRNA abundance, dicodons on dipeptides, and none of these really explained the effect at this position. We also considered RNA binding protein motifs and transcription factor binding sites. There wasn't strong evidence for either of these, but I'll note that many RNA binding proteins have unknown motifs. And so it's possible that an RNA binding protein might explain this effect. But in concordance with the prior literature and using computational predictions, we found that these 2 codons were in a highly predicted stem loop structure. And using RNA-SNP, which is a tool to predict SNP effects on structure, we found 3 variants at this position that were predicted to significantly alter structure. And so our best hypothesis for this effect is indeed an alteration of RNA secondary structure. We next looked at variant effects on translation by sequencing the total RNA and the polysomal RNA. And I'll explain in a moment what this polysomal RNA looks like. And using a similar analysis strategy, we compared the frequencies between these 2 readouts. So it's known that particular variants, especially near the translation initiation site, and even in the coding region, can alter translation. So for instance, if you have variants near the Kozak region, you might have a lower or higher initiation rate, leading to differences in ribosome load and protein abundance. And you can also have variants maybe further down in the coding sequence that can cause ribosome stalls, which can trigger RNA degradation. So there are a number of mechanisms by which variants might influence translation. So we basically took the lysates from these variant libraries and ran them on a sucrose gradient. So what you see here is a polysome profile, where as you increase in the gradient position, you have fractions with RNAs with greater numbers of ribosomes. So for instance, fraction 3 and fraction 4 would be messages that have a relatively high number of ribosomes translating them. And we subsequently looked at the frequencies of variants in these fractions relative to the total RNA. So again, we looked at silent and nonsense variants and found that in the mid and heavy polysome fractions, nonsense variants typically were less abundant. And we further identified specific variants that have an impact on mid to heavy polysome load. So you can see that many of these variants were actually up in these mid and heavy polysome fractions, indicating they might be translated better. And we wondered maybe this could be due to the alteration of translation elongation. So to get a better idea of that, we conducted ribosome profiling. So using ribosome profiling, we identified that several of these variants that showed differences in ribosome load were at putative ribosome pause sites. So you can see here highlighted with the yellow stars, variants that are located at these putative pause sites. And I'll mention that some of these pauses were either at the A, P, or E site positions of the ribosome. So while for some of these it's uncertain which of those A or P sites it is, we did note that ribosome pausing could be tied to these variant effects. We also looked at codon stability coefficients. We might expect that a codon stability coefficient, which basically measures the association between a codon and decay rate, if that is also associated with translation. As we know, translation can impact RNA decay. And we found that variants that led to higher codon stability coefficients tended to be enriched in polysome heavy polysome fractions. And we finally looked at variant effects on protein abundance. So to do this, we sequenced the input genomic DNA and genomic DNA from cells after sorting by flow cytometry. So as an example, here I'm showing a very high correlation between protein abundance and the mRNA proxy, which is read out by this iris M cherry element. You can see with the wild type non-mutagenized template, we have a very good correlation. Whereas for the variant libraries, we see a lower correlation between these two measures. And so what we did is we sorted cells into 4 different populations, where population 1 would be a relatively high RNA and protein abundance, whereas, for instance, population 3 is cells expressing variants with a lower protein abundance than would be expected given its RNA abundance. Again, we looked at silent and nonsense variants to see if there are expected patterns. So in population 1, the high protein population, we saw that nonsense variants were depleted from this population. And conversely, in population 3, the low protein given RNA, we saw an enrichment of nonsense variants and a depletion of silent variants. So this gives us some confidence that our flow cytometry method is reporting on protein abundance. And we further identified specific positions in these 2 regions where the variant might lead to low protein abundance. So there were about 12 different positions that showed many missense variants leading to low protein abundance. And if we map these onto the COMT crystal structure, we saw that many of these positions that harbored variants with low protein abundance were at positions in this protein where secondary motifs made adjacent interactions with nearby motifs. We also found that the variants nearest the SAM binding site in the catalytic center were fairly well tolerated. So many of these did not have an impact on protein abundance. And we further looked at the correlation of our protein abundance data with machine learning models for prediction of variant effect. So for instance, this DMASK score. DMASK is a linear model that uses evolutionary conservation and prior multiplex assay data to predict the effect of amino acid substitutions. And so a lower score would be interpreted as more deleterious. And so the variants that were enriched in this low protein population, P3, had lower DMASK scores. And similarly, ESM1B, which is a protein language model, where a lower value is indicative of a more deleterious amino acid, we saw that, again, the variants enriched in this low protein population had lower scores. And we further combined all these readouts and did a hierarchical clustering analysis for all the variants that had at least one significant impact in any one layer of gene expression. And we found several cases of both directional or divergent effects. So by directional, I mean that the effects go in the same direction. For instance, at this H62 position, many variants were up at the level of RNA or ribosome load or protein, whereas other positions, for instance, L136A, or these positions down here showed, for instance, increased RNA, but lower protein abundance or lower ribosome load. And we further used an unsupervised machine learning model to cluster these variants by their expression. So again, we had conducted these scans in two different regions. We had done the RNA abundance, protein abundance, and ribosome load analysis, and we can collect all these data and cluster variants by their expression across these layers. And to do that, we used a self-organizing map where variants are clustered by their kind of expression topology across these layers. And we further grouped these SOM clusters using affinity propagation clustering to arrive at 9 clusters with a substantial number of variants in each cluster. And we further looked at the composition of these clusters in terms of silent and nonsense variants. We found that this cluster 9 was highly enriched for nonsense variants. And the interpretation here is that the missense variants in this cluster then have very similar expression pattern to nonsense variants, which might indicate that they are deleterious. And even nearby clusters, for instance, silent variants in these nearby clusters might also be considered deleterious due to their proximity to this cluster 9. And so we finally asked if any of these missense variants or silent variants in these nearby clusters might impact RNA structure. So we turned to RNA SNP predictions and found that 4 out of roughly 28 of these variants were predicted to alter secondary structure. And even more interestingly, we found that the variants that were enriched in this population 3, i.e. if it has a negative impact on protein abundance, we discovered that these variants had a greater alteration of secondary structure. And so in conclusion, we had integrated multiple multiplex assays to measure RNA abundance, protein abundance, and ribosome load. And we found potential variant effect mechanisms included the alteration of secondary structure, potentially perturbing ribosome pausing, and differences in codon stability coefficients. So if you're interested in our data set, we've deposited this data to MaveDB, and we also have a paper you can check out if you'd like to learn more. And I'll just end with really the future of these multiplex assays. I think it'll be really important to integrate several of these assays so we can fully annotate variant effects on gene expression and protein function. So I would like to thank everyone for listening and also acknowledge some of our collaborators. Fritz Roth and Tina Cote have been really helpful with reagents, and Kenny Matrick and Doug Fowler for providing the landing pad cell line. And I'll just mention, I recently defended last year and am on the job market. So if you're interested in my work or have any opportunities, please let me know. Thank you.
Thanks, Ian, for the nice talk. If anyone has questions, please post this in the chat. I will start with something that I didn't understand well. Maybe you can comment on that.
Mm-hmm.
I didn't get the relationship of the polysome profile with the active translation. So
right.
I'm not sure. Yeah, if you have like an abundance of polysomes, you are going to have an active translation. Is that the link?
So this is definitely a caveat of the polysome profiling strategy. There are cases where high ribosome load does not indicate active translation. So for instance, in development, there are some examples of messages that are highly loaded with ribosomes, but not being actively translated. So that is certainly a caveat. But generally, the expectation is that the more ribosomes you have on the message, the more protein abundance. And there has been a relatively strong correlation. There's a few papers showing correlations up to like 0.6 or so with ribosome load and protein abundance. So they're definitely correlated, although there are exceptions. Yeah.
Okay, thank you.
Looks like there's some in the chat that I could.
Yeah. I'm seeing some questions here in the Q&A. Maybe I can start reading. Great talk. Why do you think many of the variants affecting expression are rare in nature?
I'm not sure exactly what this question is asking. I think certainly the common SNPs that we would observe are less likely to be deleterious because they're not under like this purifying selection. So, rare variants might be more likely to impact expression by virtue of them not being passed on because they're, deleterious to fitness.
Mm-hmm. Yeah, I have a link with this question. Could you see like any link with the previous mutations that were described, like the common SNPs for this gene?
Yeah.
Could you relate your rare variants with the common SNPs?
Yeah, so other than this SNP at the H62 codon, which was consistent and did show increased RNA abundance, we actually found that none of the other common population SNPs had an effect on RNA or protein abundance. So that's data I didn't show you, but it's in our paper. And basically, we think that the fact that we didn't find decreased protein abundance for one of these particular SNPs is potentially due to us using an express transgene that doesn't have one of the SNPs in the haplotype, which is in an intron. So it could be that, and it could also be that we are expressing this without full endogenous UTRs. So for instance, the 3' UTR in our construct is derived from the vector. And so that could be a reason why we don't see the same impacts of the common population SNPs. But we did find that the encompassing positions were in general in the same direction. So for instance, one of these SNPs thought to lower protein abundance was one of the positions we identified as being constrained. Yeah.
Okay, so another question is the evolutionary conservation of the COMT at nucleotide sequence level consistent with the effects you see?
Yeah, so we've looked at that in terms of this DMASC score, which basically reports partly on evolutionary conservation. And so we did see an agreement in terms of those positions in a global aspect, but there were particular exceptions where conserved residues were not, or variants at conserved positions didn't have a strong effect. So we did see some exceptions, but in general, it was in agreement with evolutionary conservation.
Okay, next question. Do you have an estimate of what fraction of the low protein abundance variants that can be explained by RNA translation effects, and how much is due to, for example, protein degradation?
Right. So we've looked at correlations between the RNA effect and translation effect and protein abundance. We saw stronger associations between RNA and protein abundance, and lesser associations between translation and protein abundance. As to what fraction would be due to those effects versus protein degradation, we don't have a strong estimate for that, but we would assume that many of the effects are at least on protein abundance are due to degradation. And I think your own work, Kristen, is probably meted that out, if I remember correctly. So.
Okay, and the last question here is, are these variants connected to DNA, or are they more in tune with a disease?
I'm not sure exactly what is being asked with this question. Could the attendee maybe rephrase?
Yeah, it's an anonymous attendee.
Certainly, there are CMT variants that are associated with disease. So they've been found in genome-wide association studies. As to if they are causative of disease, that's another question that I don't think has been addressed appropriately. So
yeah, I think the question is in this end, like if those variants can affect disease, but still you are describing new variants, right? So yeah. Okay, thank you so much, Ian.
I'll try to answer in the chat, I guess.
Yeah, okay.
All right. Yeah, thank you, everyone.